So why don't we start with a sound check, folks? Blue lights, cameras, action. How about a sound check? Can you hear me in the back row? Can you hear me in the back row? Lovely. We finished up what we're going to be saying about RNA transcription and processing, and a little teeny bit about whispers and suggestions about the RNA world. Are there any questions from Chapter 19 that people would like to ask before we begin discussing protein synthesis and related topics? Okay, if they occur to you later, feel free to ask them later. So this is the last chapter. We're coming up to the end of biochemistry. Not that there really is one, but it's the end of this textbook. And perhaps it's kind of appropriate that it ends with protein synthesis, because proteins are sort of the quintessential characteristic of life as we know it today, that without proteins, there wouldn't be anything like what we think of as life. And so the ability to make proteins and to make them accurately and quickly is essential to living cells. And we're going to look at that a little bit. One feature we're going to see is the fact that protein synthesis is going to be a series of repeating reactions. Just as DNA synthesis, DNA is a linear molecule, it's a repetitive motion. RNA molecule is linear, repetitive motion. Protein synthesis also will be a series of repeating reactions. In protein synthesis, there are multiple highly important roles for RNA, for ribonucleic acid. So protein synthesis is not just a protein phenomenon. It really depends on this kind of diplomatic mediator from the knowledge in the center of the cell. Some of those roles include tRNAs, transfer RNAs, messenger RNAs. We talked a little bit about their processing in Chapter 19. Ribosomal RNAs, which is principally structural but has some catalytic properties. Interestingly, it depends on energy to go forward, but the energy in protein synthesis is almost exclusively coming from GTP. This is one of the major places where we see GTP playing a role in the ongoing life of the cell. There are a few places elsewhere, but this is a big one. Synthesis alone is not enough. Proteins have to fold correctly. We'll talk a little bit about that. They have to be processed, and many of them need to be exported. We'll talk a little bit about all those different features of protein synthesis. Just to sort of mention as an aside, there are some examples of peptides that are not made by the standard protein synthetic machinery. So this is a picture here of a peptide antibiotic, and this particular one was made by a fixed array of enzymes that passed the growing peptide on to the next enzyme. So there's a separate enzyme for each reaction. All it can make is this polypeptide. It can't make others. It can't change. It's hardwired. And it's interesting and scary to see that the biochemical machinery to make things like this can be transmitted among pathogenic organisms to give them resistance to various things. So. All right. So we have to get from RNA to protein, sequence of nucleotides, sequence of residues of amino acids. Somewhere we've got to make those two come together. And that's going to be through the triplet code. You may remember that we have 20 amino acids that are routinely used in making proteins. There are a few extra amino acids that are modified after synthesis. There's some other amino acids that never get put into proteins. But in terms of synthesizing proteins, 20 is the magic number. And because there are 20 amino acids, a triplet code was the simplest code that could do the job. And we'll think our way through that a little bit. We have 4 bases in life, ATGC. Each of those bases could be present in the first or the second or third position. So it's 4 bases possible in the first position, times 4 in the second position, which is now 16, times 4 in the third position, 64 possible combinations. And because RNA is a polar molecule, there's a 5' end and a 3' end, all 64 of those combinations are distinct from each other. If there were only 2 positions and we still had 4 bases, we could only have 16 combinations. You'd have any of 4 in the first one times any of 4 in the second one gives you 16. Another feature of this code that is not necessarily something that had to be this way is it is sequential. That is, you start reading it and you keep going in sequence without any gaps. You just keep going on in a linear way to read that sequence. It's a non-overlapping genetic code. That is, you read this codon and then you go to the next nucleotide to find a codon. Francis Crick figured this out because if there was an overlapping code, there would be certain amino acids that couldn't be next to each other. And all possible combinations were found. So it's a sequential and non-overlapping. It's degenerate. Doesn't mean your mother wouldn't want you to go out on a date with it. It means that there's more than one way to say something in it. So there's more than one way to say valine, for instance. If I tell you a protein sequence, you can't tell me the nucleic acid sequence, because there's more than one way to make that protein sequence. You can make a good guess based on frequencies, but you can't make an exact back translation. Another feature about this code is that what's called the frame, the reading frame, will make a difference. And so here we have a particular RNA sequence shown here. And in one reading frame, it would read threonine, isoleucine, serine, arginine. If we had begun reading that one nucleotide over in a different reading frame, it would be proline, serine, arginine, glutamate. And if we had started 2 bases over, it would be histidine, leucine, glutamate, serine. So you can see that the frame, the place where you begin reading, makes a big difference, because that's the grouping in which you're going to take those 3 at a time. It is interesting that in all 3 cases, serine comes back into here. Maybe interesting as well that arginine gets back in here, but that sort of speaks to the fact that there's a non-random distribution of the way that the codons are distributed. This is one representation for the standard genetic code. If the first position is any of these things, and then you take the second position being U, so here we have U and U. If the third position is also U, that's phenylalanine. But you can say phenylalanine another way. You can say it UUC. So there's 2 ways to say phenylalanine. There's more than 2 ways. There's 6 ways to say leucine. And so you have clustering of the codon with a particular group of amino acids. It didn't have to be that way. UUC could have encoded arginine in terms of logic. But in practical fact, when we look at the code, it doesn't happen to be that way. If you look through this whole column, anything that has U in the second position here seems to like to be hydrophobic. Sort of striking. And over here, anything that has G in the middle position is in the hydrophilic charge range. So a certain amount of clustering of amino acid properties according to the codon sequences, although the codon sequences are completely arbitrary. Now there are 3 codons that do not code for amino acids. You can see here 3 of them, UAA, UAG, UGA. These 3 codons are going to be stop signs. When the ribosome encounters them, that's the end of protein synthesis. It's in contrast to what we saw in RNA synthesis, where there was no simple, this is the sequence that will always terminate transcription. Some of the amino acids have only 1 codon, and here is an example of that. AUG is methionine, and we're going to see that that turns out to be a very interesting codon, one with a lot of significance. Methionine is a fairly rare amino acid. You may remember it's got sulfur in it, so it's not exactly a commonplace one. Any other questions about the code before we go on? Yes? How do the what? We're going to get to that, and methionine turns out to be the start sequence, but we'll talk about that in more detail. It's a very interesting question. How do you know where to start? So we will talk about that. So there are stop codons, but there are not codons that simply mean start. So we have a code that makes an equivalency between RNA and protein, but we need machinery. It's sort of like the Chinese room where someone can read the Chinese symbol and has a book and puts up the English word without knowing anything more than that. And the machinery is accomplished or is represented in transfer RNA, tRNA for transfer RNA. And it's going to be the thing that can have something to match up to the codon on one part of itself and something that will have a competent, ready-to-go amino acid on the other part of itself. We can talk about isoacceptor tRNAs. So these are tRNAs that carry one particular amino acid, but they may match to more than one codon. So there may be several codons for valine, for instance, and an isoacceptor tRNA might read 3 of them and put valine in at each of them. So an isoacceptor tRNA allows for taking advantage of some of the code that way. How many tRNAs are there? Well, in bacteria, there are 30 to 40 tRNAs. And so you can see that in bacteria, there have to be some isoacceptor tRNAs that read more than one codon, because there aren't as many tRNAs as there are codons. By contrast, in higher eukaryotes, there can be 150 tRNAs. And this suggests that maybe there's a way to use tRNA frequencies to tune things and so forth. One of the discoveries when people started trying to put human proteins into yeast or into insect cells is that yeast and insects use a very different frequency of codons. And so one of the part of the genetic engineering was to replace the rare codons for humans, or the rare ones for insect cells, with a common one for them. So one that's common for us might be rare for them, one that's rare for us might be common in them, and so you substitute sequences until the insect cell has something that looks straightforward and simple. A tRNA is sort of 60 to 95 nucleotides long, so it's not a huge thing. Many of these bases are going to be modified. We talked about how in messenger RNA and ribosomal RNAs there are modifications. Sorry, not messenger RNA. In ribosomal RNAs there are modifications. And every tRNA, no matter what it's coding for, has 5 major features. It has an acceptor stem, and the acceptor stem is going to be where you accept the amino acid to carry it over to the ribosome. It's going to have an anticodon loop. So the 3 bases that are at the bottom of that loop will be the anticodon that matches complementarily up to the codon. And then some other features that contribute to the overall working of the tRNA. There's a D loop over here, and a T5C, which is a thymidine pseudouracil cytosine loop here. You can see here's the Phi there. Here are some modified bases here. And then the 5th feature is one that varies in size the most. The variable loop can be as small as 3 nucleotides or as big as 21, so quite variable. You can see these places here and here and here where there's base pairing and there's secondary structure there. There's helical secondary structure. But the overall thing will then pack up into one compact tRNA, so it has tertiary structure as well. And we'll see a little picture of that in a second. So this is a picture of what that tertiary structure of a tRNA looks like. And this overall shape, sort of a boomerang, sort of a chicken wire boomerang, which doesn't sound like much fun to play with, is, it'll get downright ferocious towards the end, I would think. So this is an overall shape that's characteristic of most tRNAs, almost all tRNAs, because they have to accomplish the same purpose. It's slender, it's not very wide side to side, but it's rather long. Because it's slender, several can fit next to each other in the ribosome. There are a lot of non-standard base pairs. So remember the Watson-Crick base pairs of ATGC or AUGC. being RNA. Non-standard ones, I showed you a few pictures of those of having like 2 odd ones going together or 3 doing it at once and so forth. Those non-standard base pairs contribute to that tertiary structure overall. But almost all of the bases are in some kind of stacked arrangement. They are not, for the most part, just sort of sticking out into the water or anything. They are principally stacked, although they may be stacked in a very odd base pair. The bases are out of contact with water, the way the bases in double-stranded DNA or double-stranded RNA are. And it puts the anticodon here a long way away from the amino acyl group. So it makes almost the maximum distance possible between the amino acyl group and the part that's going to actually read the sequence on the messenger RNA. And those features are all going to turn out to be functionally necessary. Questions? Mm-hmm. Okay. So how do we get the amino acid onto the tRNA? Well, there are enzymes called amino acyl tRNA synthetase, so AARs. And amino acyl tRNA synthetases can select the correct amino acid and put it onto the correct tRNA. So an amino acyl tRNA synthetase is a synthetase of putting those 2 things together covalently. And this is how it does that. It can select an amino acid. It invests ATP into it to make a phosphate group linking like that. That inorganic phosphate, of course, will get pyrophosphate, of course, will be split to help drive the reaction. So this part of the reaction then allows the tRNA to come in and transfer that amino acid onto the tRNA, and now you have an amino acyl tRNA like this. I am not sure that we fully understand the energy of this bond. My memory, and I'm ready to be corrected, is we don't fully understand why, but this is a high energy bond. And that turns out to be useful down the line. So this is where the energy is invested to put the amino acyl group onto the tRNA. And this is what that looks like in a little detail. So this is the end of the tRNA. Here is the 3' hydroxyl, and that's where we've added on the amino acyl group like that. So now we have a tRNA that has a covalently attached residue. There are some interesting things about these amino acyl tRNA synthetases. There's usually 1 amino acyl tRNA synthetase per amino acid. So there might be several tRNAs, several iso-accepting tRNAs, but they're all going to be charged by the same AARS that's specific for that amino acid. And it makes a very close contact. You can see how much of the surface of the tRNA is in contact with the AARS on this thing. I've heard someone, now, since it can charge a number of different tRNAs, it is not strictly sequence. It's obviously not reading codons, anticodon. It's reading some feature of it. And the description I've heard is that the AARS sort of sniffs the tRNA. I don't think that's technical, but it's trying to get at the idea that there's an overall aroma about it that means valine tRNA or whatever. So there's a separate domain that's going to actually make the covalent reaction. And there are some of these amino acyl tRNA synthetases that can proofread. Not all of them, but some of them can proofread. So for example, the isoleucine tRNA synthetase keeps out any amino acids that are bigger than isoleucine at the charging site itself. But if something smaller than isoleucine happened to get covalently attached, the proofreading site will hydrolyze that and make the tRNA go back and try over again. So we talked about the 3 bases of the tRNA anticodon are going to match 3 bases of mRNA. They are anti-parallel to each other. So if it's 3' to 5' in the RNA, it's got to be 5' to 3' in the tRNA. Because, as you know, whenever nucleic acids hybridize to each other, they do it anti-parallel. By convention, all nucleic acids are written 5' to 3'. And so if you want to write the sequence for an anticodon, you have to take that into account. So let's just take the codon UUC. Its anticodon is GAA. A G hybridizes base pairs here, an A base pairs here, and A base pairs here. But because we write all nucleic acid sequences 5' to 3', we had to flip it over to write it in that direction. This third position of the codon, which when you write it conventionally here is the first position of the anticodon, can sometimes wobble. That is, it may be read by more than one tRNA, can read more than one codon. And so depending on the exact structure of that tRNA, one tRNA might be able to read up to 3 codons through the wobble, or if the anticodon has modifications to put an inosine there, that may facilitate its ability to read more than one anticodon. And then this is just showing you, spelling that out in a little more detail. Here's UUC, and it's GAA reading at 5' to 3'. This is an example here of how wobble works. Here's GCU, GCC, GCA, but all 3 of them are read by IGC, right? Going back to the 5' to 3' reading. So this is a place where there's wobble in the third position of the codon. All right. So tRNAs are pretty amazing and cute. Because of the synthetases, we now have a connection between amino acids and nucleic acid sequences. But we still don't have a way to put all those things together. And that's the job of the ribosome. When we think about what the ribosome has to do, its job is to match a tRNA to a codon and make a bond form. That's its job. And all the things that we see about the ribosome underlie that underlying purpose for the ribosome. Ribosomes are one of the larger pieces of machinery in the cell. They were seen as particles in the electron microscope in the 50s, so they're big things. And this is a diagram that can show you the intimate connections between protein and RNA. So in a ribosome, there is a lot of RNA and there's a lot of protein, and those both are necessary for the functioning of a ribosome. If you look at this gray-purple one, the gray here is the nucleic acids, the purple are the proteins there. Here, they're just showing you the nucleic acids without the proteins on it to let you see how much has been filled in by the proteins. To give you a sense of how much is invested in ribosomes and in protein synthesis, it is quite typical for 80% of the cellular RNA to be ribosomal RNA. So it's a very large fraction. And the tRNA can be another, 12% or so? Yeah, so the fraction of RNA in the cell that's messenger RNA is fairly small. In general, eukaryotic ribosomes are bigger than prokaryotic, and the numbers you're going to typically hear are 80 Svedbergs for the eukaryotic ones and 70 Svedbergs for the prokaryotic. As you might guess, there are some exceptions along the way. So for instance, blue-green algae have 75 S ribosomes, and so do red algae. But in general, these numbers are helpful numbers. There are 2 major subunits to a ribosome, whether it's a eukaryotic or a prokaryotic one. So there's a large subunit and a small subunit. And in the prokaryotes, you have a 50S large subunit and a 30S small subunit. In the eukaryotes, you have a 60S large subunit and a 40S small subunit. You may notice that 60 and 40 don't add up to 80 in our math system, and that's because Svedbergs don't add. remember that Svedbergs are a measure of centrifugation. And if you put together two things together, they may well move faster than you'd expect because there's less surface area exposed. So Svedbergs don't add directly. The large subunit will have either 2 or 3 RNAs, and then the small subunit has typically 1. Okay. So the RNA in a ribosome is going to be responsible for the overall structure of a ribosome, the way it folds up and packs and so forth. Its tertiary structure, if you like, provides the overall structure. There are a lot of fairly small proteins that interact to make a complete ribosome. As you can guess, a number of those are very positively charged since there's a negative ribose phosphate backbone. Those little proteins can coat the surface, as you see here. They can also tuck inside and sort of plug holes. In the prokaryotic small subunit, there's a certain amount of flexibility to the small subunit, and then the larger one is a little more solid, sort of like a platform that the small subunit can sit on top of. Here's just an example of some of the ribosomal proteins. You can see they have no specific features in common, but they have something that's a bit unusual, which is these extended regions that are not in secondary structure and not in any compact structure. That's rather unusual and reflects the fact that they're in a rather unusual context where there's a whole bunch of phosphate backbone. So the contact between the subunits is a very important place. That's where a lot of things are going to happen. That subunit interface is going to be able to hold the messenger RNA appropriately, display it for the right things to happen and be able to move it. And it's also going to facilitate the binding of the tRNA during translation. And when we look at the ribosome in a little more detail, we can see 3 tRNA binding sites that are conventionally called A for aminoacyl. That's going to be where an aminoacyl tRNA can bind. A P for peptidyl, that's the site in which the growing peptide chain is most of the time. And then an E site for exiting after the work's been done. The small subunit, 30S in the prokaryotes, is responsible, if you like, for matching codon to anticodon. And then the 50S subunit is going to work at the other end of the tRNA and catalyze the formation of the new peptide bond. This is just showing you the 3 sites in a couple of representations. Here you're looking sort of face on with the 3 sites. And here you can see, I think sort of from the side almost, the 3 tRNAs that are bound to the subunit. And your question, it's a very good question about where it starts. Translation always starts with methionine. Whether you're a prokaryote or a eukaryote, the first codon will always be an AUG that codes for methionine. And so the first amino acid that's put in is a methionine. Now, how you pick that AUG is going to make a difference because where you picked your AUG will determine the frame for the rest of the translation. And so the fact that there's an A next to a U next to a G is not by itself enough information to tell you that that's a methionine codon. Maybe it's, half of this codon and half of that codon, right? And so there has to be more information than just the AUG sequence. In prokaryotes, there's a Shine-Delgarno sequence that lies about 10 base pairs up from the AUG. And this sequence in the messenger RNA can base pair with the ribosomal RNA transiently. It's not going to stay bound, but that's how it gets started for initiation. So the small subunit, the 16S RNA, has sequence complementary to that. In eukaryotes, by contrast, it is the first AUG you run into starting at the beginning. That tells you something about what sequences eukaryotes avoid using. That scans along till it finds that AUG. But in both cases, there's a special tRNA that's used for synthesis called the initiator tRNA. A further modification is that in the prokaryotes, the tRNA that is used at the starting site has on it a modified methionine. It has a formyl group on it. So this is formal methionine, or FMET. The FMET tRNA is the initiator tRNA for prokaryotes. Eukaryotes have an initiator tRNA, but we don't put the formal group on the methionine. Okay. All right. So how do we get started? What does it take to start translation? And we're going to start seeing here, oh, this rearranged itself. I'm sorry. This is not supposed to have been laid out like that. I wonder. Yes, it's okay on your handout. This is what drives me batty. This is produced on Microsoft. The screen is on Microsoft. Why doesn't it look the same in the two places? Oh, well. At least your handout looks good. So what's going to happen? Well, the ribosome is going to have to pick up several things to begin with. Let's see, how should we begin this? Let's start thinking about, here we have the 30S subunit, the small subunit. We've got messenger RNA over here. And we have an initiator tRNA that's in an interesting complex. You notice here that the initiator tRNA, the FMET tRNA, has IF2. So IFs are initiation factors, IF2, and a GTP bound to it. And if we have all three of those things present, the small, free, empty, solitary, small subunit, free message, free FMET tRNA with its initiation factors, we can put together a complex like this. So this is the first step towards trying to assemble a functional ribosomal complex. Why doesn't it, the initiator tRNA is going to sit at the P site, the peptidyl site. Why doesn't it start at the aminoacyl site? Well, that's because initiation factor 1 has the job of preventing that. It sits there at that site to make sure it doesn't get, the incoming tRNA doesn't get mixed up. Once we have this complex, the next thing that has to happen is we have to add on top of that, like the top to a hamburger bun, the 50S subunit that's going to sit on top of the small subunit and make a complete complex. And when the large subunit comes in, it releases IF2, and you notice that we've hydrolyzed the phosphate on the GTP to GDP. So this is one of the places where the GTP hydrolysis is an important part of the process. Until it's hydrolyzed like that, IF2 is not available to do its job again. Okay? So that's the fairly simple story of how prokaryotes initiate translation. In eukaryotes, it takes more stuff. There are on the order of around 9 initiation factors. There's an initiator methionine tRNA that's a specific tRNA for the initiating site. It's going to have the same codon as the other methionine tRNAs, but it's a specific tRNA, and messenger RNA. Some of the things that need to be present are cap binding protein. You may remember the 5-methylguanosine cap at the beginning of eukaryotic messages. So there's a cap binding protein that recognizes that that cap is present. The cap is telling the cell, this is a message, it is intact, or at least it's intact on this end. There's a helicase, because you can imagine these large messenger RNAs can have secondary structure. You might want to undo that. And poly-A binding protein. Remember, the poly-A is at the 3' end of the messenger RNA. And poly-A binding protein does, in fact, bind to poly-A, as its name tells you. But it's part of the initiation complex because it interacts with the eukaryotic initiation factor 4G. And so to get a functional initiation complex, you have to have an intact cap at one end and an intact 3' poly A tail, and the poly A binding protein brings it around till you have a circular template ready to go for the process. The small subunit, the 40S subunit, needs to use ATP to scan. So it starts at the earliest place it can and is going along, and that scanning process is expensive. It costs ATP. And there is a bit of context. So in other words, this is the AUG that you want. That's the one that's going to be methionine. But this sequence in front of it is part of what makes that AUG the magic AUG that starts the frame. And so you can see there is some sequence context that helps the ribosome recognize that this is the right AUG. In analogy to the prokaryotes, IF2, but in this case EIF2, meaning eukaryotic initiation factor 2, delivers the methionine tRNA to the P site again, the peptidyl site. And like in the prokaryotes, it takes GTP hydrolysis to free the EIF2 for the next one. And now we have a complex that the large subunit, the 60S subunit, can bind to and make a complete ribosome. So initiation is a bit more complicated in eukaryotes, partly because the message structure is more complicated, partly because eukaryotes seem to do everything in a more complicated way. But the underlying things are still in common. The need for IF2, for GTP hydrolysis, for message, for tRNA, and so on. But the other features are what make it have an additional level of surveillance for intact, good-looking messages. Prokaryotes grow so fast that if they've got a dismal message, they can throw it away. But eukaryotes have a slower rate of doing stuff, and so you want each message to count. And this business of having the cap binding protein and the poly A binding protein is part of this process of both protecting the message and of telling the protein synthetic machinery that it's intact. Yes, question up there. Right. Right. If you notice here, this is the, so as you're saying, the Kozak sequence is upstream of this sequence? Well, is this the Kozak sequence here, or is it removed just by other nucleotides in between? Okay. I will defer to you on that. I thought that was somewhat under discussion, or has the discussion been closed? No, it's like kind of Kozak sequence, so that's the first Kozak. Right. Okay. All right. So, okay. So we've got an intact ribosome now that is ready to elongate. We've got the first one in there, but we need to then add others. And what we're going to see is, in common with RNA synthesis and with DNA synthesis, it will be a cycle in which certain reactions are repeated again and again. And so for each cycle, a new aminoacyl tRNA has got to enter and bind at the A site, at the aminoacyl site. For that to happen, there is a factor that helps deliver it to the correct site. And in prokaryotes, it's called EFTU. EF for elongation factor, and TU because it's temperature unstable. There's a little reasoning behind it. EFTU binds GTP, like a number of other things we're going to see in the course of protein synthesis. And EFTU does not care which tRNA you are. It binds to all tRNAs. And so this is part of the role for that T5C loop. You remember that there were the two side arms. There's the aminoacyl, the acceptor stem where the aminoacyl group was going to form, and there was an anticodon. The other two loops are partially involved in things like this, where EFTU can then recognize that it needs to be carried in. And this is showing you, here is the EFTU, and here is the tRNA. Okay. So who gets to sit down and stay for a while? Well, the tRNA that is going to be held there is going to be selected by its affinity for the codon at the A site. So it's a question of the base pairing between the anticodon and the codon. And if you take a look at the structure of the ribosome with and without its tRNAs, here is an example of how tightly packed together the 3 tRNAs are on the ribosome. So you've got the 3 codons on the messenger RNA, and the tRNAs actually have to kind of be splayed out a little bit apart from each other to fit 3 of them on that stretch of messenger RNA. And so it has to make that appropriate contact. There's a little bit of a actual distortion, if you like, in the backbone of the messenger RNA, and the ribosome can then examine whether or not that's a good fit at that spot. So, all right. There's an interesting feature in protein synthesis. So we've seen extensive attention to proofreading and error correction in DNA. There's a bit of effort to proofread in RNA synthesis. There's proofreading in protein synthesis as well. And one aspect of it is what's called a kinetic proofreading. And so if you have a good fit, an appropriate fit, one-to-one, a perfect fit codon to anticodon, that fit is communicated by conformation to EFTU. And EFTU will then hydrolyze this G to GTP and go away. If, on the other hand, an aminoacyl tRNA comes in and is not making a good fit, there will not be GTP hydrolysis, and it will go away. It will come off again without the next step having happened. The error rate for protein synthesis is about 1 in 10,000. So it's a pretty accurate process, but it's not as accurate as either RNA or DNA synthesis. And that's because this is the end product. It's not going to get amplified. A mistake in a protein at this point is either going to make no difference, or it may lead it to it being denatured and therefore not fold correctly, in which case it'll be eaten up by the protein cleanup machinery. One thing to think about, if it's one error in 10,000, you start to think about how big proteins are. It means that a significant number of proteins have at least one error in them, but in many cases they can be accommodating. Okay. So this is showing you the elongation cycle more diagrammatically. Here you have a ribosome ready to accept tRNA at the A site. If you have a correct tRNA, you're going to get base pairing that's effective, that's transmitted to the EFU, which hydrolyzes the GTP, comes off, and now you're ready for the next step, which is called transpeptidation. If, by contrast, you had an incorrect EFTU come in, or tRNA come in, like this, it'll hang around a little while, and then it'll just come off and won't have spent any GTP in the process. So one thing to take away from this is it costs at least 1 GTP for every amino acid added. We took 2 high-energy bonds back at the aminoacyl tRNA synthetase, or synthase. Here's another high-energy bond that's needed for the process. So how do we actually get the next, how do we actually get a new bond? How do we get a peptide bond? This is catalyzed through the 50S subunit, the big subunit. So the small subunit is playing a role at holding on to the messenger RNA, sort of kinking it a bit so it can fit the tRNAs and helping to match it correctly. The 50S subunit is going to catalyze the new bond. And what that looks like is something like this. So you have now in the P site the peptidyl tRNA, that is the tRNA that has a peptidyl group attached to it. And in the A site you have an aminoacyl tRNA, which is a tRNA with just a single aminoacyl group on it. And the large subunit is going to catalyze the formation of a bond so that this amine group is going to attack over here, and it's going to in effect vacuum up or pick up growing peptide chain. And after that new bond has been formed, you're in a situation like this where the P site has in it only an empty tRNA, an uncharged tRNA, and for a transient period the A site has the peptidyl tRNA. It's not going to stay there very long, but for a little while the A site has the new elongated chain. Proteins are synthesized from the amino terminus to the C terminus. So just like RNA and DNA have a direction to synthesis, protein does too. And interestingly, the catalytic site on the large subunit is RNA. So at perhaps the most important part of protein synthesis, the actual catalytic moment is done by RNA. It has to be held by the ribosomal proteins, but the catalysis is an RNA. And this is an example of something we talked about way back in the first part of the course, proximity and orientation. That part of what it's doing is it's holding these two things close to each other, and it's contributing the, not merely being close, but the orientation of them relative to each other. Any questions about that before we move on? Okay. All right. So that's the transpeptidation reaction, but then we need to open up the aminoacyl site and make it available for new tRNA. And so that event is called translocation. So this reaction is transpeptidation or peptidyl transferase. Then we need translocation to change where the growing chain is. So this new peptidyl tRNA slides over into the P site. The empty tRNA that used to have the peptide chain and is now empty slides over into the E site, the exit site. And the mRNA is slid down one codon, keeping the peptidyl tRNA in frame with it. So you have to sort of imagine the messenger RNA and tRNA sliding as a unit, if you like. This also requires elongation factors. We talked about EFTU. This is elongation factor G, or EFG. And interestingly, EFG looks like a mixture of EFTU with a tRNA. So it's a mimic, it's a molecular mimic for EFTU when it's in complex with tRNA. And so here's an example of the structure of EFG. It's possible that it actually dumps the peptidyl tRNA to get it to move down. And this is also going to cost us a GTP for it to happen. So it cost us a GTP to load an aminoacyl tRNA in. It costs us one to translocate it, but it didn't cost us one at the moment we were making a bond. And maybe that's a good place to stop until Wednesday.